my pleasure to have introduced Dr. Hope Rugo from the University of California in San Francisco.
Great, thanks very much for selecting this for the press conference. And I'm actually presenting on behalf of a collaboration of I-SPY investigators and supporters. And this is the first efficacy results from the multi-arm I-SPY 2 trial. I-SPY 2 is a unique phase 2 trial that's designed to screen a series of novel agents in combination with standard chemotherapy in the neoadjuvant setting. Patients are randomized to receive a novel regimen in combination with standard chemotherapy or standard chemotherapy alone, with the endpoint of pathologic complete response. This trial design will accelerate the process of identifying drugs that are effective for specific breast cancer subtypes and thereby reduce the cost, time, and numbers of patients needed to get effective drugs to market. We are reporting today just one of 7 current experimental arms of I-SPY 2, and I'll note that I-SPY 2 includes agents now from 5 different pharmaceutical companies. This shows you a summary of the study plan in its most simple form. Patients are randomized after meeting eligibility criteria to receive weekly paclitaxel or weekly paclitaxel with a series of investigational agents or regimens and followed by standard AC or doxorubicin and cyclophosphamide chemotherapy. That is then followed by surgery. The primary endpoint of I-SPY 2 is assess response rates as measured by PCR of investigational agents in combination with standard therapy versus standard therapy alone. And for the purpose of this trial, PCR is defined as no residual invasive cancer in the breast or lymph nodes at the time of surgery by standard histochemical techniques and using a specific method for analysis called the residual cancer burden, published by Fraser-Simmons. The I-SPY 2 trial design is novel and includes adaptive randomization as the key for enrolling patients. It uses a pre-specified and automated algorithm, and randomization probabilities are updated as the study proceeds by signature and based on MRI and then PCR results. The algorithm triggers the decision to graduate when 60 to 120 patients are enrolled in the experimental arm. Randomization is based on performance of regimens within biomarker subtypes or signatures, and there are 8 biomarker subtypes based on hormone receptor, HER2, and mammaprint high, and then ultra high. Mammaprint high and ultra high are based on the median cut point of mammaprint for I-SPY 2 eligible patients, and the percentages of distribution in the different signatures today for the entire trial are shown here. This is all of the signatures. I'm going to just show you this more today for the purposes of brevity, the signatures that are important for this arm of I-SPY, which include all of the HER2 negative patients, so that's one group. And the second group are the triple negative, hormone receptor negative and HER2 negative. And the third group are the patients who are hormone receptor positive and HER2 negative, so 3 different signatures. A drug can graduate, or a regimen, when there is an 85% predicted likelihood of success in a randomized phase 3 neoadjuvant trial that includes 300 patients with PCR as the endpoint. When a drug graduates, accrual to that arm stops, although accrual to the master study continues without change, except for that arm is dropped. When the 85% success threshold is defined based on the response of each patient's tumor assessed by MRI scans over time, and then recalculated when the pathology results are available from definitive surgery on the basis of PCR. This includes all patients in that arm, and then patients who are randomized concurrently to the control arm. This is a cartoon showing the I-SPY 2 adaptive trial design. Over the course of the trial, we incorporate information about response based on imaging that's updated then with the final pathology, and that informs the randomization assignment for the next patient who comes on study. This allows us to learn and adapt from each patient as we go along, and each new patient benefits from information obtained from the prior patient. In addition, this allows us to add and drop agents as we go along based on success of each agent or regimen. Patients are eligible for I-SPY if they have tumors of at least 2.5 centimeters, and then they undergo a core biopsy of their tumor as well as an MRI scan. In order to be eligible, you have to meet certain stratifying biomarker shown in this table. Specifically, patients whose tumors are hormone receptor positive and HER2 negative, as well as mammaprint low risk, are not eligible to proceed to the randomized portion of the trial. Patients who are eligible are then randomized and sign a second consent that is specific to the arm that they will be treated with. The experimental arm that we're reporting today includes Veliparib, a potent oral PARP inhibitor, and Carboplatin, a chemotherapy agent. For this analysis, patients were adaptively randomized to receive Veliparib at 50 milligrams twice a day for 12 weeks and Carboplatin at an AUC of 6 every 3 weeks for 4 doses, with weekly paclitaxel and then followed by AC, as shown in this schema. The concurrently randomized control patients received weekly paclitaxel followed by AC, and enrollment, as I showed you earlier, was only open to patients with HER2 negative disease, and patients are eligible to graduate in the 3 signatures I mentioned earlier, all HER2 negative, hormone receptor positive, HER2 negative, or triple negative. 72 patients were randomized to receive Veliparib and Carbo with standard neoadjuvant chemotherapy, and 71 patients are evaluable. 62 patients were concurrently randomized to the standard regimen, and 44 have HER2 negative disease. The I-SPY2 Bayesian model estimates the probability distribution of PCR rates in each signature, which is a new way to look at trial results. The actual PCR rates are biased by the adaptive randomization and are not calculated. This shows you how we estimate the probabilities for pathologic complete response. The distribution of PCR rates for the control rates is in blue, for the novel agent, or in this case regimen, and standard treatment is in red. And then the mean of distribution is the point estimate for PCR and is referred to as the estimated PCR. These are the results from the trial. This shows you the estimated PCR rate for the all HER2 negative signature. What you can see here is that there is considerable overlap between the two curves. The estimated PCR rate is 22% for control and 33% for the experimental arm. But the probability of success, in other words, the probability that Veliparib and Carbo is superior to control, is high at 0.92. When we look at this further, we found that this was all due to success in the triple negative signature. As you can see here, the estimated PCR rate is 26% for control and 52% for Veliparib and Carboplatin, which corresponds to a probability that this experimental regimen is superior to control of 0.99. In contrast, the patients with HER2 negative hormone receptor positive disease, there was considerable overlap in the estimated PCR rate and a probability of superiority to control of only 0.28. The format for the summary of results reporting that I'll show you next is listed here with the estimated PCR rates by signature, the likelihood that the drug is better than control for a given signature, and then the probability of success in a 300-patient phase 3 trial based on our estimated PCR rates. The data is shown here, and as you can see, Veliparib and Carboplatin has graduated in the triple negative signature. There is a 52% estimated PCR rate for the experimental arm compared to 26% in the concurrent control, with a 99% probability of superiority of control and a 90% predicted probability of success in a phase 3 trial. In summary, the I-SPY2 standing trial mechanism, a novel trial design, efficiently evaluated this agent combination in a biomarker-defined patient subset. We identified a biomarker signature drug pair for Veliparib and Carboplatin on the basis of a modest number of patients. Veliparib and Carboplatin has graduated with the triple negative signature, the subset recommended for this regimen's subsequent development. I didn't show you the toxicity, which was moderately increased as expected, but it was well managed with dose reduction and delay. The trial was not designed to evaluate the individual contribution of Veliparib and Carboplatin, but the effect size is large for the combination. I-SPY2 is a biomarker-rich trial. Additional response predictors are under evaluation, and specifically my colleagues, Dr. Olapade and Vantvere, are evaluating markers of DNA damage, including BRCA status. And of course, there are multiple members that are critical to the success of this multi-center collaborative trial. Importantly, Drs. Laura Esserman and Don Berry developed this trial and are in the audience. And Dr. Esserman has worked for many years with our collaborators in order to obtain collaboration between multiple pharmaceutical agencies, as well as the FNIH Biomarker Consortium and our funders, to develop this novel, forward-thinking trial. We also, of course, have multiple site principal investigators, our project management office, working group chairs, and agent chaperones, and the patients who participated in this trial. Thank you.
Judy Paris with the Journal of the National Cancer Institute. Are you saying that when a drug graduates with X percent predicted likelihood of success in a phase 3 trial, that the endpoint for that phase 3 trial is also going to be PCR? And if so, you have reason to believe that that's going to be acceptable to the FDA?
So yes, the plan is the randomization is designed, and with this threshold of success, with an endpoint of PCR, a similar endpoint to this trial, in a randomized 300-patient phase 3 trial 1-to-1. And as you know, a lot of work has been done with the FDA recently on evaluating drugs for accelerated approval based on their response in the neoadjuvant setting. How this plays out in the future for other drugs after the accelerated approval of pertuzumab remains to be seen. Clearly, there needs to be a plan for outcome data that can correspond to the PCR rates in order for these drugs to obtain final approval, but it is the hope that this will be a mechanism for accelerated approval.
Dan Keller, Oncology Times. A 300 subject phase 3 trial seems fairly small. Is that because you've done a lot of the upfront legwork and you can recruit faster and get the results out faster? What's the rationale for only, say, around 300 patients?
If you're able to select the group of patients whose tumors are most likely to benefit from a specific treatment, you can actually enroll less patients in the trial and still have a high chance of success in proving that the experimental arm is better than the standard. So the whole goal here is to identify the right patient group to treat and regimens that are effective, and then treat them in a smaller trial that can give you positive results.
Carolyn Helwig, Gasco Post. So when deciding which patients would go on to which drug, how molecularly driven was that assessment? I mean, I know you used MammaPrint, but that is really a test of recurrence risk, isn't it? Or can you explain? I mean,
well, I didn't show you all the different signatures, but basically patients are divided into these different groups based on hormone receptor status, HER2 status, and MammaPrint divided into 2 categories within high risk. And then based on those signatures, they're randomized. And of course, that's changed as the trial goes along and enrolls patients based on response.
but it still seems fairly general to me. I mean, in this day and age of genetic expression.
Well, you know, it's a very good question, but we don't know. That's one of the points of the trial, is to actually look further into these groups. So, for example, this combination graduated in triple-negative disease, but there's tissue available both at time of treatment and as patients went to surgery, and a number of different biomarkers will be evaluated to try to see whether or not we can further define the group that benefits the most.
Okay.
But I'll point out that we've done a lot of biomarker studies on trials that have accrued to date and have yet to identify biomarkers that help select the patients that are most likely to benefit. We're better at finding prognostic biomarkers. And in this kind of prospective trial design, we're most likely to be able to identify those groups.
Excuse me, Lynn Peterson with Trends in Medicine. So about 10 days ago, another participant in the I-SPY 2 trial announced top-line results, but no details. And can you offer anything on that?
I can't at this time. Really, we just have the details that are in the press release from Puma regarding neratinib. That data will be presented at a scientific meeting next year.
Thank you. Peter, we're ready for the next speaker.